Hello everyone. Today we'll talk about a problem which came in CSIR UGC NET June 2025 regarding Luciferase reporter assay, and in this context we can also learn something about gene regulatory network. So at first what we need to know what is Luciferase assay. So in Luciferase assay what we do is we try to look for the activity of the transcription factors, for example, and the strength of the promoters. So here our objectives are to look for whether a protein is a transcription factor, or and also you have, like, if you have a transcription factor, how efficiently can the transcription factor drive the expression of the target genes. So how do you do that? So in order to do that, you take a transcription factor, for example, you take a target gene promoter. So let's say this is a target gene promoter, and afterwards there is the, so in normal context you should have its CDS, and afterwards there will be some poly signals or something like that to stop the transcription, right? So in this case, so in place of the promoter of that particular gene, what we do is we actually incorporate the luciferase sequence. So here there will be, so we'll be adding the luciferase CDS. So whenever the promoter will be active, it will drive the expression of the luciferase, producing the luciferase protein. So we will get luciferase here. Now, when we have a transcription factor, let's say for example, which has been found to be helpful in uh, more than basal expression, then the transcription factors, for example, is binding to the promoter. Transcription factor is binding to the promoter, and it will drive the expression of the luciferase more than its basal level. So for example, in this context, they have taken, so that they have taken into account the activity of the two promoters A and B. So you are uh, talking about two gene promoters. For example, there have been we are studying the promoter of gene A and gene B. Upstream of a luciferase reporter in response to a transcription factor X in a cell line was studied. Luciferase activity was measured in cells following overexpression and RNAi silencing of transcription factor X, and compared to control, which is the native transcription factor X level. Luciferase enzyme activity is tabulated below. So basically what you have taken, you have this promoter of the gene A and you have the luciferase downstream, and you try to look for how much luciferase is being expressed. So let's say I have this pGene and then I have this luciferase CDS like this. So for example, if I have this transcription factor in a native state, like native expression level, so this transcription factor, what it is doing is its basal expression level for promoter gene A. We don't know whether the transcription factor is actually driving any expression of the PG, like whether it's activating or repressing the promoter of gene A. So that's what we have to check. So the basal level is 9, but when you are overexpressing the transcription factor X level, like overexpressing the transcription factor X, or the transcription factor X supposed to be go up, the promoter activity is going down horribly. Also, we can see that the RNAi silencing also leading to the same effect. So here we can say that it's something which tries to keep a basal activity. So transcription factor X is required to maintain a proper basal activity. So these are sort of an example, like these are sort of examples of gene regulatory networks where there will be some transcription factors, which transcription factors will be really important in maintaining the basal expression. For example, if this TF-X is being expressed in a low level, it will help in the expression of the gene A. However, when it's being expressed in a lot, the gene A will be the gene A will be repressed completely. The gene A expression won't be happening. So these sort of examples can be seen in some developmental biology context. There is a really good example in some pair role genes, gap genes in Drosophila. So I would like to comment to you guys what is the gene, what are the genes which shows, like what are the transcription factors which shows this sort of gene regulatory network. So we have one more promoter gene B. So in this promoter gene B, what we can see that the basal expression level is 2, and when we are overexpressing it, like it's going up. So it's like saying that it's not important in the basal expression, but it's important in its of like it tries whenever the transcription factor X is being expressed, the expression of the gene B will be higher compared to normal. Whenever you are also silencing the transcription factor X, it just keeps the expression level in its basal level. So that means that transcription factor X is required to overexpress the gene B. But in the absence of transcription factor X, the basal level, like the promoter activity doesn't change. So out of these three statements, we can say the transcription factor X is needed for the basal activity of promoter A is correct. Transcription factor X is not needed for the basal activity. Instead, it's required for its hyperactivity. So this statement is wrong. Transcription factor X functions both as a positive and a negative regulator for promoter A. This statement is also correct. So we can say the statement 4 is correct.